Good morning, everyone, and welcome to our gene delivery seminar series for December, our last one for this year, 2025. We have Kat with us today, and Kat is going to tell us a little bit about SPARTA Biodiscovery and the cool things that they've done. Kat Saunders is a senior application specialist at SPARTA Biodiscovery. During her PhD, she worked on the development of the SPARTA, that is single particle automated Raman trapping analysis technique, to apply to a range of nanomedicines, and has since joined the company SPARTA Biodiscovery, working with clients and industrial collaborators to unlock the potential of SPARTA for their samples. We are very excited to have you here, Kat. And yes, go for it when you're ready.
Great. Thank you for the introduction. And I just wanted to say thank you to the Vector Builder team for the invitation, to give this seminar today, and thank you all for your attention. So it is my pleasure today to talk to you about next generation analytics and how we can unlock these label-free insights at the single particle level. So I know that I'm talking to the right audience when I say that nanoparticles are evolving rapidly to produce safer and more efficient therapies. However, in order to achieve this, these nanoparticles are becoming increasingly complex systems incorporating novel carrier components, cargoes, and targeting moieties. When we're preparing these nanoparticles, the ideal case is that all of our particles are totally identical in terms of size, cargo loading, and carrier composition. However, in reality, we will have heterogeneity in these samples, so there will be different subpopulations of particles with variation in all these different parameters. For example, my slides are being a bit funny. For example, we might have larger particles with more cargo or smaller particles with different lipid composition. And this heterogeneity is combined with this increasingly complex multi-component systems where there can be variation and heterogeneity in properties, including the vehicle components. So for example, in LNPs, lipid nanoparticles, I mean things like the lipids, in addition to the cargo, the nucleic acid, and also the service moieties. And therefore, to really capture this information, so this spread in these different properties, and to really understand these nanoformulations, it is key to use analytical methods which look at these properties on the single particle level to really get a clear picture of this inherent heterogeneity. And so it is to answer these challenges that we developed the SPARTA Aegis 1 instrument. This is a benchtop analytical instrument, which has been designed to be accessible for scientists to gain deeper understanding of their nanoformulations. Some of the key features that we offer are single particle resolution by performing Raman spectroscopy on individual nanoparticles, and in my next couple of slides, I'm going to explain exactly how that works and what information that gives. There is also minimal sample interference, meaning that the measurements take place in nanoparticle suspension, they're label-free and non-destructive. Additionally, Raman spectroscopy is inherently multi-parameter, so you can simultaneously measure the cargo, carrier, and surface components on a single particle simultaneously. Additionally, the technology is compatible with a range of biological nanoparticles, and this is all wrapped up in an instrument which has simple and reproducible operation with a dedicated analytical software. So before I really dive into the science, into the technique and its applications, I did just want to take the time to thank all the scientists who've contributed to the studies that I'm going to showcase in this presentation. And for a little bit of background information, the SPARTA technology, it was developed in the group of Professor Dame Molly Stevens at Imperial College London, who has now recently moved to Oxford University. And alongside a range of peer-reviewed papers, the technology was patented, and this now forms the basis of the company SPARTA Biodiscovery, which was spun out in 2022. And so as was mentioned in the introduction, for my involvement, I did my PhD in the Stevens group, working on developing the SPARTA technology, and it was a few years ago that I transitioned to the company. The data that I'm going to show was generated both within the academic research group and also the company setting. And alongside, all of these wonderful scientists who are listed here, I also have to thank our strategic partner Sartorius for helping us to bring this technology to market. So how does SPARTA work? Well, we combine two Nobel Prize-winning techniques, specifically Raman spectroscopy and optical trapping, and this is combined with a proprietary trapping algorithm. So Raman spectroscopy is this first technique, and this has been an experimental science for around 100 years. In Raman spectroscopy, we shine a laser onto the sample. There is then an energy exchange between the incoming photons and the molecular bonds in the sample. These photons are scattered off. And this amount of energy exchange is dependent on the specific molecular bonds. So when these photons are scattered and we analyze this energy exchange, we can work backwards to work out what are the chemical bonds and what is the chemical composition of our sample. So what the information that you get out is in the form of a Raman spectrum, and you'll see there are different peaks here, and each one of these different peaks corresponds to a different chemical bond that is in the sample. So by analyzing these peaks and the relative intensity, we can gain really a lot of detail about the sample, kind of this snapshot, kind of simultaneously, including the carrier composition, the sample purity, drug loading, and functionalization. And we want to do this on the single particle level. So what we do is we then bring in our second Nobel Prize winning technique, which is optical trapping. In optical trapping, we take a very tightly focused laser beam and we shine this into a solution of the nanoparticles. This very tightly focused laser beam creates an optical trap or a confocal volume of less than half a femtoliter in the nanoparticle suspension. When nanoparticles diffuse into the volume, they feel a trapping force, and then they're stably held in this very small volume until we decide to release them. So in this way, by combining Raman spectroscopy, optical trapping, and the proprietary automation algorithm, we have designed a non-destructive, label-free, single-particle chemical profiler that allows us to build up a population-level overview of the chemical composition of a nanoformulation with single-particle detail. Some of the advantages of this are that we measure a holistic view of the particle population by measuring several parameters at once, and the same technique can be applied to a range of different nanoparticles. So this includes lipid nanoparticles, viral vectors, polymer particles, and hybrid particles. So what parameters can SPARTA tell us specifically? Well, because we measure the full Raman spectrum of individual particles, we can measure these multiple parameters simultaneously with single particle resolution. This includes nanoparticle composition, such as cholesterol and ionizable lipids or protein content, targeting moieties, such as proteins or sugars, the degree of aggregation, and the amount of loading, for example, of RNA per particle. So now, with all of those promises, I'm going to introduce some SPARTA data to explain how we can access this information. What I'm showing you here is the Raman spectrum from a pristine particle. So this is an average spectrum. It's made by averaging all the individual particles that are measured in a sample. So you can see there's a solid line, that's the mean line, the average line, and the shaded region is the standard deviation. We can visually see there are some clear peaks, and these correspond to specific bonds present in this nanoparticle. And the nice thing about Raman spectroscopy being an experimental science for 100 years is that all these peaks are very well documented. So as a sanity check, we can also compare these peaks to databases, and they will not substantially shift in position. So we can, for example, take these peaks and assign them to the base nanoparticle. What you'll see is that if we take this particle and add on a coating polymer, then there are clear additional peaks that we can visualize. For example, here around 1300 and 1450 wave numbers. So we can assign these peaks to the polymer. Again, they can be checked against databases in prior literature. What you'll see is, again, kind of in this iterative way, if I take this system, so I take the nanoparticle plus the polymer plus the nucleic acid, we kind of have these same peaks in the nanoparticle and the polymer, and we additionally have these peaks from the nucleic acid. So similarly, we can assign these to the nucleic acid specifically. Okay, so this is the average spectra. But something, you could measure this on a wide variety of Raman instruments. However, of course, we really want to break this down into the single particle information to really understand the heterogeneity. So what I want to take now is example of performing loading analysis with the SPARTA technology. And what I'm going to do here is I'm going to take two examples of two different loading conditions with the same nucleic acid. So to do this, I can do a univariate plot. So this is where I look at the nucleic acid signal, and I'm going to compare an unloaded control to the two loading conditions. So what you'll see on this univariate plot is that there are all of these dots. Each one of these represents an individual particle that was measured. And the higher the dot is on the y-axis, the more of this nucleic loading acid signal that we're detecting. So basically, the higher the loading, the higher it is on the y-axis. And there are a few different ways that we can, just to explain the plots as well, we also have the violin, that's showing the distribution, and the line in the middle, that is the mean line. And there's a few different ways we can interpret this data. So one of the things is to look at the ratio of loaded to unloaded particles, and we can do that based on the nucleic acid signal. So what I can do is that I can set a threshold on the unloaded control, a statistical threshold, and then we can compare the percentage of loaded particles. So you see that for both of these conditions, it's very high. It's at 96 and 98%. And just to note, you'll see there's quite a spread in this data. So I should say this was a very short type of RNA that was used here. So we're expecting there to be kind of tens to hundreds with a reasonable distribution of particles, rather than the more kind of discrete, empty, full populations you might expect kind of from the longer RNAs. So this is kind of showing us the ratio of loaded to unloaded particles. I can also change the position of this threshold to look at the average amount of one loading condition compared to another one. So this is a 50% threshold on the Na1 condition. And what you can see is that actually 79% of the Na2 particles have a higher signal. So what we can see is that, this Na2 condition, it led to more particles with more loading. So we'd say this is kind of the higher loaded, the optimal condition. So this is if you want to do kind of the more straightforward analysis. What we can also use is the fact that, of course, we have measured all of this different chemical information. So we can then start to move to bivariate plots where we're adding in extra parameters to understand the relationship between different components. So, what you can see here is a bivariate plots. This is really showing you the relationship between the polymer signal, so your coating, and your loading. And, you can start to see here in terms of subpopulations forming and also, kind of distribution of these different, these different components. So these are, the three main types of analysis that we would use when we're looking at the SPARTA data. We'd look at the mean spectra to find the peaks of interest and to try and understand the sample chemistry. We then use the univariate and the bivariate plots to examine the relative amounts of components and then identify the relationships between these different components and also for subpopulation analysis. And this is, kind of our workflow, and I'm going to show you how we apply this with a couple of different case studies and how you can then, kind of integrate this into your analytical workflow. So what I'm first going to start off with is some data which actually isn't our typical opener, and I'm going to talk about contaminant analysis. So what we found is that this is something that we're starting to really have a lot of conversations about more and more, given, the recent challenges in the cell and gene therapy field, specifically relating to CMC. So whilst this is, of course, a very complex and far-ranging issue, we do believe, and also based on some of the conversations we've been having, that we can get to some way of addressing these concerns through introducing kind of more advanced analytics, more detailed chemical understanding earlier in the pipeline. So specifically, due to the complete chemical picture that SPARTA provides, we can identify both biological, leachable, and solvent contaminations. So the first example that I'm going to show you here is where we can identify these biological contaminants. So this is a collaboration with Hansa Biomed, which is a company that produces EVs, or extracellular vesicles. EVs are biological nanoparticles that are made up of lipid, protein, and nucleic acid components, and these are released from cells as a signaling and kind of, a signaling mechanism. And they've been receiving kind of this increasing attention for diagnostics and therapeutics. However, one of the really key challenges with the translation of these EVs to the clinic is their purification. And in particular, it's really known that it's very, very challenging to kind of separate, kind of these EVs from other biological nanoparticles of a similar size and density, known as lipoproteins, because these very, very commonly co-isolate with EVs during the most common purification methods. So what we have here with these EVs is a case of a very heterogeneous sample where it's really important to understand the different particle populations that are present. And so this is really where, it kind of comes into the fore to use these single particle methods rather than relying on the bulk analyses where you'd get, one sample, one kind of averaged result. What I'm showing you on the left-hand side here, this is a bivariate scatter plot. What we're doing here is we're simply plotting the lipid signal against the protein signal. So each one of these dots, again, it's an individual particle that was measured, and what you're seeing are two different samples, blue and green. These and the colors represent the EVs derived from different sources. You'll see the different shades. These are just 3 technical repeats from these 2 samples. And from this analysis, we can identify 3 particle subpopulations. There's the desired product, which is kind of in that bottom left-hand corner. And then there are also 2 different types of contaminants. One which is very, very high in protein, which we can link back to kind of more protein aggregation. And then there's the contaminant 2, which we're kind of seeing as the lipoproteins. And in this case, what we clearly saw from this analysis was that, one of the EV sources, the source in green, this resulted in much, much higher purity than the other one. So we had much, much more of the product with this particular EV source. And so, you can imagine this kind of analysis. In this case, it was, very useful for identifying, where these EVs should be coming from in terms of source, but it could also be used for kind of comparing and selecting between different production or purification processes. So that is biological contaminants. And another type of particulate which can be present in the samples is plastic particles and leachables. So actually what we found was in these same EV samples, we could detect kind of small plastic particles. And these are leachables which can be introduced during the production process, ironically during some of the purification steps. And now, because the Raman spectroscopy is chemically specific, we could detect not only the presence of these particles, but we could also chemically identify them to this specific plastic and kind of identify the source of these. And so having really this deep understanding of the formulation and kind of tracking these leachables, kind of what process step they were introduced in, it also, led to a bit of tweaking of the purification process and eventually, their removal in the final product, which was kind of a great result for our collaborators. So those are 2 examples of contaminating particles, which can be in the sample. Of course, if we're making particles, kind of these synthetic particles, for example, LMPs or polymer particles, what can also be present in the samples is solvents. And we work quite extensively in the lipid nanoparticle space, so I can show you briefly some of the work we've done with kind of ethanol detection and quantification. So now, because ethanol is in the solution, we can quite readily make a calibration curve of ethanol in the buffer at different concentrations. So what you can see on the top here, this is the main ethanol peak across 3 repeats. And you can see on the bottom, this is then resulting in a highly linear calibration curve. So by analyzing this, we get a kind of a limit of quantitation of 0.1% volume over volume. And now, of course, the proof is in comparing to real samples and established techniques. And in this, we also get very excellent agreement. So you'll forgive me, this is ongoing work, so I can only share one number with you at the moment. But using this method and correcting for sample dilution, we calculated a SPARTA ethanol concentration of 10.2%, which is within 2% error of the gas chromatography measured value of 10%. So that's been a little snippet of, kind of what SPARTA is able to show you beyond what you're, let's say, expecting in your sample. But, what we can also show is, some very much more positive things. And this is, really kind of getting new insights into, kind of what we're expecting to see in the samples. So I've got a couple of case studies to show. And the first of these, this was work carried out at Imperial College London in the group of Professor Molly Stevens, and this is work that was led by Dr. Adrian Nayer. So the purpose of this study was to develop a malaria therapy. Now, the mode of action for this therapy was to bind to invasion receptors on the parasite surface. So by blocking these receptor sites, it then prevents them from infiltrating red blood cells, because this is really, these parasites, they enter the blood cells, and then this starts the life cycle of reproduction and release. So to achieve this, the researchers use a targeting polymer, which is known to bind to the receptors. However, this polymer by itself was not very effective, kind of just at blocking them. It worked much better if it was kind of incorporated into a nanoparticle. So therefore, really to make sure that there was, it was kind of incorporated into a nanoparticle, some different lipid components were introduced. So what we end up with is then, you want these lipid and polymer hybrid nanoparticles to get this to really work effectively. However, this is kind of a very novel, complex, multi-compart nanoparticle. So this also then requires very careful analytics to determine how these components are spread between the particles and to understand, are really true hybrid particles achieved. So when making these particles, there are, a whole suite of analytical tools available. So here what was used was dynamic light scattering, and this is a very standard technique, kind of to determine the size distribution of kind of more homogeneous samples. And there was also some small angle scattering done, and this is a technique which can tell you about particle morphology, including kind of size and shape. And the main conclusions from these techniques were that, these particles were made of around 100 nanometers and they had, they were kind of like lamellar particles. However, these are, as I put here, these are both bulk techniques. So they're looking really at the ensemble average. And so it's always important, to then employ kind of single particle techniques as well. So what was also included was some cryo-electron microscopy. And, this is, really kind of visualizing the single particle morphology. So it's showing, there's some quite interesting vesicle structures, which was kind of then revealed. And so, from this quite intensive analytical suite, there is really a lot of information that has been gained about the nanoparticle sample. But actually, inherently, this hybridization question, it's a very, very detailed chemical composition question. On a single particle level, do I have my lipid and my polymer components, kind of on this one particle? And actually, these techniques, even though they give a lot of information, they can't really answer this question of, do we have true hybrids? So this is now where SPARTA was employed. So what we looked at was 4 different samples. So these were 2 samples containing polymer, and these are these 2 samples on the bottom here, and 2 samples without polymer. What was also compared was the influence of incorporating a PEG polymer. So this is kind of for stability. And so this is the sample, the second from the top and on the bottom. And so in terms of analyzing data, what we can use is the same workflow as I showed you before. So we first look at the mean spectra, which is these over here. If you remember, this is the average spectra of all the individual particles that were measured. And what we can do is we can look at, what are the differences between these spectra. We can also cross-check them with databases. And we can identify that there is kind of a strong PEG peak at around 840 wave numbers. There is lipid signal in kind of all of these samples, and then there's polymer signal at 1610 in these bottom 2 samples here. So now with this, now that we've identified this, we can actually pick out, this specific polymer peak and then start to look on the single particle level, kind of with the univariate analysis. So what you can see here on the right-hand side, these are the controls. These are with no polymer. And then on the left-hand side here, these are kind of the two hybrid or hopefully hybrid particles. And again, so there's this average line. This is what you'd really get, kind of from the bulk methods. And then each of these dots is an individual particle. And what's very, very clear from this analysis is that actually both of these hybrid formulations were showing clear polymer signal kind of across all of these particles. So what we could do from this, quite simple analysis was then we could confirm that there was hybridization occurring and these particles were made of both lipid and polymer components. What's also quite interesting, and one of the reasons that we also really like this single particle analysis is that we could detect a difference in heterogeneity kind of based on the formulation. So kind of as a side observation here, you can see that actually in the formula without PEG, this was a nice tight distribution, but actually including PEG kind of increased the compositional heterogeneity of these particles. It increased the spread of this polymer signal that we were seeing. And what I'll say is that, in this case, this was kind of the first study just to really understand hybridization. So, this was just a nice observation. But in the next stage of this study, this is really where, this came more into play. So as a follow-up, the same researchers, they're interested specifically in targeting severe malaria. And what they were doing with this was some formulation optimization. So with this, what they were doing was increasing the relative amount of polymer in these formulations. So what we can do is a similar analysis, and we can kind of compare the amount of polymer signal for each of these formulations. So you'll see on the x-axis here, there's these 4 samples. There's one with no polymer, just lipid only, and then this is kind of increasing the initial amount of polymer that was added. And what you'll see is that as we're increasing this polymer amount, the amount of polymer incorporated is increasing up until a saturation point. So for this, I can just look at these mean lines. And this is what you'd get really from the bulk techniques. However, when we're looking at the single particle information, so kind of the individual dots, what we can see is that actually, even though the averages are the same of these last 2 formulations, there's quite a big difference in the sample quality. So you see, actually, there's a much, much higher spread in this final kind of highest formulation compared to the other one. So in other words, what we've done here is that they've added so much polymer that it's actually really destabilized the formulation. It's caused a lot more heterogeneity in the composition. And so, it's with this information that it allowed the scientists to really kind of select this middle condition and to kind of to move forward with the biological assays and the product development. So just to confirm, conclude this, SPARTA really provided the single particle composition information, which was required to understand how successful this hybridization was. And then it also kind of allowed the researchers to optimize their polymer concentration, ensuring that the best candidates were then taken forward to, kind of the testing round and the development of the products. So that case was looking at polymer-lipid hybrid nanoparticles. What I now want to highlight is a recent study by one of the teams at AstraZeneca, led by Eliza Lazaro Ibanez, and this was looking into LNP EV hybrid nanoparticles. So in this study, the scientists, they were using extracellular vesicles to tap into their safety profile and targeting ability. However, these EVs, they're very difficult to load with mRNA. So to improve this loading, the team was using kind of the standard mRNA LNP system, and then they were combining these to make these hybrid EV LNPs. Now, because this is a very, very complex system, the scientists, they were using a wide variety of analytical techniques. So this kind of included Western blots, DLS, ribogreen electron microscopy. And these provided, really a lot of important information on the particle size, morphology, and loading. However, what I'll say is that, in a lot of cases, these either required fluorescent labeling or they only gave, kind of single parameter information. So only, size, for example. And this is where the group really used SPARTA for kind of the detailed chemical information from a single snapshot. What I'll say is that these guys had really a lot of fun with the analysis and they went very, very deep with it. So I'm not going to go cover kind of all of what they did, but I just want to show you like a few of the highlights. So first, I want to look at kind of some of the chemical information they pulled out. So one of the first things that they wanted to do was to compare the protein content of the base EVs, the base LMPs, and the hybrid EVs. So what you can see here, if I take just this left-hand graph, you'll see that there's quite a big spread of protein in the EVs, this protein signal. This is what we expect because these are, again, they're kind of these cell-derived biological nanoparticles. There's kind of a lot of inherent heterogeneity here. And then these next 2 conditions, so the second and third condition, these are 2 different types of LMP. And as we'd expect, there's kind of very little protein signal here. What's very interesting is that then, kind of when combining these LMPs and EVs into the hybrids, what you also see is that there really is like a lot of heterogeneity in both of these conditions. So this is kind of showing that, in terms of like the final sample, the final formulation, you probably have some particles which are kind of still EV and still pure LMP, as well as quite a broad spread, quite a heterogeneous spread kind of these hybrids. And this is confirmed looking at, a couple of different protein signals, and also, when looking at the lipid signals as well. So you kind of see the inverse here. The EVs, they kind of are scoring a bit lower on their lipid profiles, but the LNPs, of course, are scoring very highly. And again, there is really like a real spread in terms of the hybrids and this lipid signal showing that, there was some successful hybridization, but it's really, there's quite a lot of heterogeneity in these samples that were made. And so, this was then really interesting in terms of kind of the information you could pick out. So you can look both at, let's say like a generic protein or lipid signal. And you could also really pick out kind of very specific kind of signals here. So, what you can do is to validate both the fact that you've, kind of made a hybrid formulation, and then also really to kind of understand the compositional differences between these different formulations. So the final case study that I want to show is a pure LMP one. This is also coming out of Imperial College London, led by Dr. Hannah Barriga. And this is looking at stimuli-responsive drug delivery systems, so stimuli-responsive LMPs. And specifically, this is a question about monitoring changes in lipid composition. So the scientific background is that there is a particular enzyme phospholipase D or PLD, and when this cleaves the PC head group to form a PA head group. So what you can do is that you can take these kind of responsive lipid nanoparticles, incubate them with the enzyme, and you'll see a change from the PC to the PA head group. And you can then use this for kind of a controlled release application. So just to show you, kind of how SPARTA was used here, we can take the same workflow as before. So we can start off by looking at the average spectra. And what you can see is that the cholesterol amount was changed in these particles. This was kept constant, and then the PC or the choline amount was changed. So what you can see is we've got in the average spectrum, the cholesterol peak is very stable, and then the choline amount is kind of changing on average as we're kind of switching over from the PC to the PA. And then you can see, if I'm breaking out into the single particle information, we've got kind of the univariate plots over here. And you can again see, there's the average line is decreasing, but the whole population is decreasing as well. And then, of course, what's really interesting to do here is to look at this, in terms of kinetics. So looking at a time course analysis. So now having, kind of related the signal to the amount of change in the choline to cholesterol ratio, we can kind of then track this over time. So what the researchers then did is that they took these nanoparticles, they incubated them with different amounts of the enzyme, so low, medium, high amount, and then they measured this for just over an hour. So what you can then see if we do this is, each one of these dots again is an individual particle, but now the x-axis is kind of looking at this over time. What you can see is that actually this interchange between the head groups, this was very, very dependent on the amount of enzyme that was added. So as you'd imagine, the lower amount of enzyme led to kind of slower kinetics, whereas the kind of the longer, you had a much shorter changeover with kind of this higher amount of the enzyme. And so this then really, it led you to understand kind of the influence of the enzyme, and then also, a bit of this kind of formulation tuning as well to really tune the kinetics of this changeover. So those were the three case studies that I wanted to show you. And now I just want to kind of, to summarize by saying that, again, what SPARTA really gives is these multiple parameters from a single measurement with single particle resolution. So what we measure with the instrument is the nanoparticle composition, the functionalization, drug loading, and the amount of aggregation. And this really then allows kind of this deeper product understanding of, kind of these really, really essential questions such as formulation optimization, storage condition effects, batch analysis, cargo release, and, kind of response to stimuli. And just to really summarize, you get this kind of very comprehensive analysis with the instrument. You can measure, kind of the whole particle in a single snapshot. So that's both the kind of internal loading, the composition, and what's on the surface. And you can really get this kind of multi-parameter, multi-component measurements out. So with that, I wanted to, thank you for your attention. And I'm happy to answer any questions.
Thank you so much, Kat. That was really excellent. Very cool technology. Um, I want to remind everyone that if you have any questions, please put them in the little thing that says Q&A, and I will be able to convey them to the speaker. We have one question. How do you see this approach being used to study or even optimize gene delivery vectors, maybe particularly in terms of like tracking cellular uptake or predicting successful transfection events in real time?
Yeah, it's a great question. And so I know that this presentation was very much focused kind of on the more synthetic nanoparticles, so LNPs, but this technology, it is also compatible, kind of with viral vectors. So I would say that if I'm looking back to, let's say, my this part, what we find at the moment is that really the batch-to-batch analysis and the purity is a really important thing, which are kind of our current customers are finding a lot of use for. So, there's always this question of, if you've isolated something, especially kind of these gene vectors, these viral vectors, how much of your um, yeah, like how much of your sample is kind of empty, full, or some other type of contaminant. So this subpopulation analysis is something which we really have found a lot of benefit for. And we are starting to look for, let's say it's always the aim is to replace these very kind of time intensive, weeks long kind of efficacy cell studies. It's not something we can claim yet, but it's something which, we're very interested in what we can tell with this detailed analysis.
I have another question which may be related. I feel like you partly answered it, but how do you see the applications of SPARTA for viral vector analysis, particularly, such as distinguishing full versus empty capsids? Yeah, or is the system currently better suited for non-viral nanoparticles?
Yeah, it's a great question. So, yeah, I would say that's one of the things which we definitely do is, kind of looking at the viral vectors. So, one thing to clarify is that we don't do, it's not like proteomics. It's not that we're distinguishing between different proteins. What we're really seeing is kind of protein content, nucleic acid content, and contaminants. So I'd say, by analyzing those, you can really gain kind of really a lot of information. So it's, the empty, full, and contaminant. And that subpopulation analysis is, I'd say, yeah, kind of the real benefit that we see and we bring.
That makes sense. Thank you. We have another question. How feasible do you think this is for widespread incorporation into the drug development pipeline in terms of time, cost, and regulatory approval?
Yeah, it's a great question. So one measurement takes around an hour at the moment. So, this is the kind of the timescales that we're working on. I would say at the moment we tend to focus more on the R&D and kind of process development side of things. But it's something which, as I say, our customers, they have been having a lot of fun kind of integrating into the analytical workflows.
Thank you. Is given, sorry, is Sparta able to correlate the chemical fingerprints of nanoparticles with downstream biological performance, maybe such as uptake or even transgene expression?
It's a great question. It's something I can't speak too much on at the moment because we don't have anything public. But let's say it's something which we are working on.
Okay,
Um, yeah. So I mean, we found very much that, heterogeneity and purity go a long way to explaining a lot of, let's say, kind of biological effects.
cool.
But I
Thank
can't say
you.
too much more.
No, that I totally understand. Thank you. I think this is our last question. Oh, never mind. Can Sparta analyze RNP loading CRISPR-Cas9 in lipid nanoparticles?
RNA loading, I guess.
Yes. I mean, for CRISPR, it would be the gRNA too, right? Protein and the, yeah.
Yeah, it's a good question. So with lipid nanoparticles, we tend to more focus on kind of the lipid composition. This is because when you're measuring a lipid nanoparticle, what if you think about the composition of it, it's usually only around 1% of it is the RNA. So we tend to get much more signal from the lipids, but it is something which it is feasible. But we actually tend to find that, looking at the lipid heterogeneity and the lipid spread, you can actually already tell really a lot of information.
Oh, thank you. Given SPARTA's ability to monitor changes in particle compositions, do you know how the nanoparticles interact with biological media or cellular components over time?
Yeah, it's a good question. So we have done studies on kind of protein corona formation. So, this idea that you take the particles, you add them into biological media, and the proteins kind of stick to them. So we have had quite a lot of success with doing these studies. And I should say the system is compatible with measurements in, so not full serum, but up to about 10% serum. You can kind of do measurements in this and start to look at the kinetics of that.
Great. All right, thank you. I think those were all the questions that we had. If anybody has any questions last minute, go for it. If not, thank you very much, Kat, for being here, taking the time to walk us through this really cool technology. And thanks everybody for attending this talk. Happy holidays in advance, and we will see you in January.